Hi everyone, I'm Lin Zijin, a second-year PhD student. My project is to design clinical trial with a model to quantify capsaicin adverse event. Today I'll answer three key questions. First, how to model clinical adverse events. How to use modeling to inform the design of a clinical trial. What's a good strategy for quantifying clinical risks? Capsaicin, as we all know, is a widely used oral chemotherapeutic agent, 5FU project, which can be used in colorectal, breast cancers, and other solid tumors. However, the hand-foot syndrome is the most commonly reported adverse event caused by capsaicin, which can cause the dose reduction and treatment interruption. It's also difficult to predict the hand-foot syndrome onset and trajectory. This figure was reported in HANA 2009. They reported about 600 patients using the same dosing regimen. The aim of this paper was to calculate the probability changes over time. Due to the limited data set on grade 2 and grade 3, they merged these two grades. From this plot, we can conclude that the probability changes substantially over time, suggesting a dynamic and heterogeneous hand-foot syndrome time course. This heterogeneous time course can be explained by the higher variability in capsaicin pharmacokinetic. During 2005, they collected 40 patients, performing normalized concentration profile. From this plot, we can see the capsaicin concentration varies by 4 orders of magnitude, from 0.05 to 50 micromole per liter. Hence, we proposed a model-based clinical trial design method to identify the minimum sample size and minimum observation duration. We set 3 steps. First, we need to select a reliable dynamic modeling. In my project, I used the ordinal modeling reported in HANA 2009 and used the same dosing regimen as HANA 2009. The patients took capsaicin in 3-week cycle with 2 weeks on and 1 week off. After experiencing hand-foot syndrome grade 2, the patient will receive a reduced dose at 50% or 75%. Then we can simulate the ordinal modeling under different scenarios, such as different sample sizes and observation duration. Then we can fit the ordinal modeling to this data size, and finally, we can get the sufficient sample size and observation duration. So the first question is how to model clinical adverse events. I will use the ordinal model here. The ordinal model can help us to quantify the cumulative probability over time. In HANA 2009, they introduced the log of odds to calculate the probability. So this is to calculate the grade 0, and this is to calculate the grade 0 or 1. Then they use specific equations to represent this log of odds. So this is a complete formula for calculating grade 0. This is for calculating grade 0 or 1. Specifically, we can see a term K times Q. The Q here is the cumulated dose, which is calculated by the kinetic pharmacodynamic modeling. Which is the kinetic pharmacodynamic modeling is the simplification of population pharmacokinetic modeling. What needs to be noted that the Q here is not a real pharmacokinetic value. It is used to mimic the higher individual variability in the pharmacokinetic. and K is a constant value which represents the elimination rate. They also incorporate creatinine clearance as a covariate to show the effect of renal failure. From this model, we can get the probability of each grade occurrence and grade transition number. The second question, how to use modeling to inform the design of a clinical trial. We will use the simulation refitting process here. By simulating the ordinal modeling under different scenarios, we can generate varying sample sizes and observation durations. Specifically, sample size of 120 were randomly sampled from 400 patients to reduce the random effects caused by simulation. The observation duration of 24 and 18 were curtailed from the end of 30 weeks. After fitting the ordinal modeling to this simulated dataset, we need to answer this question. Can parameters be fitted reliably? After that, we can identify the minimum requirement for the new clinical trial. Throughout the whole process, I use the same evaluation criteria to identify reliable parameters. So this is the relationship between relative standard error and the sample size. We can see obviously from this plot, the relative standard error decreases as the sample size increases. For the sample size of 400 patients, 10 out of 12 parameters achieved the relative standard error below 30%. Hence, we consider the minimum sample size was 400. I conducted the same test on observation duration. The relative standard error didn't differ very much across different observation duration. To obtain a more informative dataset, we set observation duration to 30 weeks. So for the final question, what's a good strategy for quantifying clinical risks? The answer is phase 1 dataset. So we can apply ordinal modeling to phase 1 datasets, and I'll take Micron 1998 clinical trial, for example. In this phase 1 clinical trial, they collected different types of adverse events with different grades and different dose level. So this is a multiple doses clinical trial. They also reported the pharmacokinetic profile of capecitabine and its metabolites. Hence, in the phase 1 dataset, we can get both the adverse event and the pharmacokinetic information. After that, we can predict the probability of each grade and predict the clinical risks. For now, I can answer these 3 questions. We can use the ordinal model to predict the clinical adverse event. We can use the simulation refitting process to inform a new clinical trial design. And the phase 1 dataset can be a better choice for quantifying clinical risks. Thank you.